What is your kind name?
Sir, my name Khushi Bajaj.
खुशी. तो बेटा किस टॉपिक पे आपने प्रोजेक्ट प्रिपेयर किया है?
Post fertilization in plants and ecosystem.
Okay. What is post fertilization event?
The events which are occur after the sexual reproduction in plants, they are
Okay.
known as post.
So what events used to take place after fertilization?
Seed formation, fruit formation.
Okay. Seed formation, fruit formation, endosperm development.
Embryo.
Okay. What is the difference between true fruit and false fruit?
True fruit occurs from ovary and false fruit occur from both ovary and thalamus.
Very good. Can you just give one example of one false fruit?
Strawberry.
Strawberry. And what is the difference between GFC and DFC?
GFC is the grazing food chain, whereas DFC is the detritus food chain. In GFC, there are grasses, or in DFC they occurs from dead materials like saprophytes.
Very good. Now just tell me, what is productivity?
Productivity, it is the time taken to perform a task.
Not the time. It is the amount of biomass produced by the living organism like plants, okay, with help of sunlight. And what is the difference between primary productivity and secondary productivity?
Primary productivity is the rate of amount of biomass produced in per unit time, or
Very good.
it can be measured as gram per meter square.
Very
per
good.
year.
Very good.
Or the secondary productivity, it is the rate of amount that is of biomass produced by the consumers.
Very good. And there is some relationship between GFC and DFC. What is that?
Somewhere GFC depends on DFC food chain for
Okay.
their survival, as they are the dead or decaying matter.
Okay.
Or the GFC, the animals or plants in the GFC like grasses or grasshoppers may be depend on the saprophytes or the detritus.
Okay. And there is one more relationship, that many of the members of GFC and DFC are common. Okay. And which experiments you have done?
Isolation of DNA,
Okay.
mitosis of onion root tip, and frequency density.
Frequency, density, and which spottings you have seen?
Ascaris, Plasmodium, Entamoeba histolytica.
Okay. Any other?
Testis, ovary, and blastocyst.
Can you just tell what is the outer layer of testis called?
Tunica albuginea.
Very good. Tunica albuginea. And which stain we use to add to stain the root tip? Take your time. Acetocarmine. Very good. Why we used to squash or tap that slide?
To spread the cells.
To spread the cells. Very good. And why we used to take only the tip part? Why not some back part of that root?
Because it contains the meristematic.
Because it contains meristematic cells. It means you have studied it thoroughly. And why we used to use the glass rod in that experiment of DNA extraction during the spooling process?
Because the glass rod is also polar and the DNA is also polar, so it helps to precipitate.
Very nice. Very nice. Best of luck.
Thank you, sir.